Hi all, I am Dr.A.Sherlin Rosita, Assistant Professor, Department of Bioinformatics, Bishop Weber College. Today we will explore how molecular modeling tools helps in designing a drug through various computational drug designing tools. So in combat with the rare and expected disease in today's world. So the impact of disease raised with 9 million deaths in 2021 as per the World Health Organization out of 100 billion population. So number of disease continue to rise in each of the statistics. Why there is an increase of disease? Have you ever wondered, aren't sufficient drugs available, and why there is a lack in drug availability? So increase in the rate of disease incidence urge for the search of new, novel and potential drugs with less side effects. So coming to the pathway of identifying a disease, isolating a protein and preclinical studies until the FDA approval before the investigational new drug application has been filed, till the FDA approval, the development begins in the decision made to put into a clinical phase by developing an assay. Until the marketing comes, it will take nearly 12 to 15 years, and we need to spend across $800 million to bring a drug to a market. So this is going to be the tedious process of using all these assays in identifying a new drug for the upcoming pandemic diseases. So through the drug discovery process of generating the hypothesis and the sources of molecules from natural, synthetic and as well as from biological sources, we are in need of to develop a hypothesis, and we need to perform some assays, we need to screen, and we need to go with the initial hit of compounds. And coming to the drug discovery process lane, and these drugs which has been screened must be put into a secondary evaluation, analysis leading to a chemistry and efficacy test. Next, by going with the third process, we need to develop that candidate and bring it to a market, and all around this will take around identifying a drug and finding a molecule, testing its efficacy, whether it harms to a human or whether it involves in the in vivo method of any other organism. So it will take around 12 to 15 years and with a cost effective manner. So we are in need of a quick and effective drugs to search for new against a particular target, reduce the drug discovery processing period and the reduction in the process cost. So the only way we need to focus on this is computer aided drug designing. It's a quick and a need for an effective method to search for new drugs in combating all those rare diseases. So this drug discovery pipeline based on the pharmacological, molecular biology and automation, medical and analytical chemistry study, we are an inclusive of this bioinformatics approaches. So the basic bioinformatics approaches, we are in need of genomics, proteomics, molecular biology, information technology, databases applications, computational, and the major part is computer aided drug designing as the main source of combating and finding a new drugs for all the potential targets. So the computer aided drug designing is nothing but it is a specialized discipline that uses computational methods to analyze the drug and receptor interaction. The drug is nothing but the medicinal compounds, either it can be a natural, synthetic or from biological sources, and the receptors, the membrane bound receptors, which is responsible for the disease mutation. It depends on the bioinformatics tools, applications and databases. The goal of CAD, that is computer aided drug designing, is not only to find the ideal drug, we need to find the lead compounds that has the specific biological property and need to optimize that ligand by screening its bioavailability process. So coming to this drug discovery process that brings in the computer aided drug designing of cost saving must be efficient, time to market and insight into the drugs has to be a major role while identifying and predicted using this bioinformatics approach. So this pathway which has been earlier applied in all those wet lab techniques has been applied in the computational approach with the help of by identifying a disease using genomics, proteomics And high throughput screening of more than 1 lakh compounds in a single database is possible against a target protein, and through virtual screening, and we can predict its bioavailability, whether the drug has been orally bioavailable for an organism. And combinatorial chemistry by screening the vast variety of compounds by bringing into a market with a good piece of compound. And the molecular modeling tools to improve its better efficacy and higher affinity, we can apply the molecular modeling tools to make the drug in an efficient way. And finally, the drugs which has been proved with great efficacy can be taken into an in vitro and in silico studies. We can avoid all those trial and error method in wet lab, and the best drug candidate can be posted into a in vitro by saving the time and the cost. So this can be done with the help of, while identifying a disease, the disease can be identified once, and its target identification by through structural determination. The structure determination can be confirmed with X-ray crystallography and NMR, and through various computational aided tools, modeling tools, we can design the drug, and the biological assays can be performed with the help of the computer based screening, and the chemical drugs with the help of combinatorial chemistry, and finally these drug target can be put into a clinical trials phase. The only way we can come through this process in an easier and efficient manner with the help of computer-aided drug designing. So these are the key areas in CAD research. One is virtual high throughput screening and the sequence analysis. So if there is no structure available for the particular target protein, we'll go for homology modeling, and again it's for similarity searches. We can optimize the drug based on our efficiency, and physicochemical modeling and the oral bioavailability of the drug can also be performed. Next, the methodologies used in CAD. There are two types of drug designing available. One is the direct design, another one is the indirect design. So while moving for the direct design, the disease structure, that is the gene which is responsible for that particular disease, is known. So this is called as structure based drug design, that is the spatial target is known. And in indirect design, the ligand based, so we know some folklore medicines. For example, when we go for some Ayurvedic medicine, we know that it is responsible for some fewer diseases, but we don't know how that particular ligand has been targeted that disease. So in that time, if the structure of the target is unknown, we'll go for ligand based drug discovery, in which the ligands, that is the orally bioavailability of ligands, which has the specific biological role, whether it can target that disease genes through systems biological approach. So coming to the structure based, it relies on the 3D structure of the biological target. So using this target, the drugs have higher affinity and higher selectivity. So when the structure of the disease is available, that is the gene is available, so that gene has been put into a higher affinity. So when that particular gene has a high affinity towards the drug, so that drug has a good binding potential binding active site which is present with an interaction, so that the electron sharings with protein and the compound is highly affinity and selectivity. So only that drug candidate can be fit into a particular target with the help of lock and key model using molecular docking. So in this way we can go for a designing of a potential drug candidate. So how to determine the protein structure? When the structure is available, we can move with a 3D database called protein data bank, and if the protein structure, that is which is responsible for that disease, is not available, we'll move off de novo designing, that is new designing by identifying its interaction sites and homology modeling. And after that, when the structure has been confirmed, that is 3D structure of a protein, that is 20 amino acids when it combined to a protein, when it is confirmed, so we can identify its interaction site, we can evaluate the structure and synthesize the compound and make interact with the lead compound. So the drug candidate, so from where do we observe and get the drug candidates? Through natural, that is from the plant extract or animal, biological engineered through recombinant technology, synthetic, that combinatorial chemistry, and by synthesizing the drug, and we can also source the drug from semi-synthetic like antibiotics. So taken into a human or an animal, what method and what traces will remain, whether it has any side effects or not, can be checked with the help of the computational tools. So a drug should have, when the synthesized drug, we must go into a safety measures, and it must pass through few parameters like safety, efficiency, stability, solubility, and finally it must have some novelty. So in order to get a ligand, so earlier we have seen that structure as determined from the protein data bank or through de novo designing. The ligand, we can take in the ligand from the PubChem or the drug bank. These are all the structure databases, or if the structure is available, that is in the raw format, and we are not able to download it from the databases, we can go through some drawing tools like ChemSketch, ChemDraw and Marvin Sketch, and we can upload the structure for our analysis. So once the drug has been confirmed, the drug must be orally bioavailable. So if it is seems to be have a drug-like properties, it must pass through the Lipinski rule of 5. So Lipinski is the author who framed the 10 rules. The drug must have not more than 5 hydrogen bond donors and not more than 10 hydrogen bond acceptors, and a molecular weight under 500 Daltons, and the octanol partition coefficient, that is log P, is less than 5. So when a single drug passes through these parameters, it is orally bioavailable and it is safe for an organism to intake. So once it has been confirmed, in order to evaluate the structure, we can go with the interaction with the protein studies. And how does this drug work? So this is a basic model of enzyme substrate complex. So the substrate will bind to an enzyme that is with a correct direction, that is into an active site. It is seemed to be like a lock and key model. So when it has a high affinity and high selectivity, so it will bound into an enzyme substrate complex. This is called a lock and key model. So when the gene and the substrate is nothing but a ligand, so when it is bound together, when it has some interactions and electron sharings in a good manner with the help of the potential energy surface, and these bound to an enzyme substrate complex. So when the protein structure, if it is known, we can derive from the PDB. If it is not known, we can go with the homology modeling. And this is the active site prediction, where a drug actively bound to an enzyme in a particular site, and it has a role of sharing of electrons, and with the help of bonding and non-bonding interaction, the drug will show its potentiality and bioavailability through the interaction studies. So we can use this. One of the tool which I have mentioned is Q-site finder to finding the active site in enzyme. And this is a lock and key model, often we call this a molecular docking. Here the compound has been interacted with the protein, which seems to be a better drug. So with the help of the algorithms, a molecular docking is performed to lock the structure. We can design and identify the keys. So this is the best example. A compound costanoid is bound with a ligand protein called PPR gamma. Costanoid is a natural compound and which is bound into a PPR gamma, which is responsible for the obesity, and obviously it will lead to diabetes. And through the images we can see the costanoid has a better binding affinity with the PPR gamma in diabetes patients. So hence the electron sharing and non-bonding interactions has been presented here. We can say this compound has a better binding affinity and potentiality with the PPR gamma responsible for the diabetes. So coming to the end, to the conclusion, I just want to explain you that development of refined pharmacological activity that is better for the parent molecule, and with the help of these computer-aided drug designing school, more potent and selective bioavailability drugs can be produced. So current clinical use or late stage of clinical development can also be applied. So for example, in the recent scenario, we have came across this corona virus, and the immediate need of drug discovery has been a very tough task for us. So while moving to this pipeline of drug discovery, bioinformatics and computer-aided drug designing plays a vital role in repurposing a drug for coronavirus with the help of all those respiratory syndrome FDA approved drugs. They have applied it in the computational methods and developed a new drug model for eradicating this COVID virus. So with the help of this, development of target-based therapies is possible, and it helps scientists to be more focused on drug discovery approach with less time and cost effective. Thank you.